It's indeed a matter of delight for me to be present in this symposium. This is my first close interaction with this gathering. And I will say 2 sentences about the history. The birth of the modern medicine has taken place from these systems of medicine. 200 years ago, it was the Greek system, now we call Unani, which was adopted in toto by the then called allopathic system of medicine. With the discovery of morphine from Papaver somniferum in 1805, the whole ray of pure molecules started coming. And it was Tibia College in Delhi that gave a lead to, from an Ayurvedic drug, Rolfia serpentina sarpgandha, isolation of reserpine. The mark which was discarded by the scientist of Dr. Sadiqi at that time and Ajmal Khan, that was taken to Switzerland, and that reserpine was characterized there. And the very first drug in the modern medicine for hypertension, Serpacil, came to the world market. I think that's the history of the Indian systems of medicines. When we talk of the diseases, the WHO has recognized about 120 diseases. In spite of all the scientific advances that have been made in the drug development and drug discovery, in the pharmaceutical sector, I'm sorry to say that 1/3 of the diseases have a cure. Remaining 80 diseases, they are managed symptomatically. How much great challenge is before the traditional systems of medicine to fill the gap. In traditional systems of medicine, there is a single drug therapy, there is a classical formulations. Fortunately, India is one of the richest biodiversity nations so far as medicinal plants are concerned. And we have been using since ages the drug armamentarium, where 75% are constituted by plants. And then we have the minerals, metals, marine products, and animal products, combinations of these. When it comes to the question of single drug, there had been innumerable problems right from the nomenclature of the drug itself. Since we had the different systems of medicines, and because of slavery of the hundreds of years, these systems are suppressed like anything. And after independence, I must salute the government of India, having given a due recognition, full patronage, full financial support for these systems of medicines. Now, these plants, when they were considered to be used in the Indian systems of medicine, there was a nomenclature problem. And that created a lot of confusion in the identity of the plants. Up to 1960 or 65, there had been a lot of confusion. The very first pharmacopoeia, Ayurvedic pharmacopoeia, came in 70s with a very small number of monographs, but very dedicated work, the first chairman, Sir Karnaran Chopra, of that pharmacopoeial committee. With this, an exercise started of evolving a system by which we could prepare monographs for the pharmacopoeia. Today, with the full support of the councils and the Ayush, Department of Ayush, we have successfully accomplished publishing 6 volumes of single drug for Ayurvedic Pharmacopoeia of India, 4 volumes of Unani Pharmacopoeia of India, covering more than 500 plant drugs, where the nomenclature has been clarified, clear-cut botanical nomenclature with the correct identification, and certain parameters of quality have been given. So far as the compound formulations are concerned, there are certain procedures which are involved, and those procedures, as laid down in the scriptures, needed elaborated standard operational procedures, what we call as SOPs. Because unless the raw material is standardized, the process is standardized, we cannot get a finished product which is of consistent quality. This exercise was attempted about 5 years back to take up the compound formulations. Now there is a complex situation in compound formulations. The number of ingredients varies from 5 to 10, 10 to 20, 20 to 70. Very hard exercise, but a humble attempt has been made in laying down the SOPs of about 150 compound formulations and their quality parameters, whatever at the disposal of the scientists were there, were laid down. Still the question arises whenever the question of acceptance of these drugs is concerned, quality is the major issue. Why this quality is a major issue in spite of the fact that we have produced pharmacopoeias for standards? I think, and the Drugs Act, which was, the Chapter 4 was introduced after 1960 in the Drugs and Cosmetic Act with respect to the rules and regulations of the Indian systems of medicines. It is the execution and implementation of those rules and regulations in the states and even at the central level that is totally lacking. And we must admit our weakness. Whatever questions are raised are because of the non-implementation or very poor implementation and execution of those rules and regulations. Of course, Department of Ayush is doing a lot, but still there is a need to strengthen the drug control administration for the Indian systems of medicine, both at the state level as well as the central level, and also the strengthening of the analytical laboratories for maintenance of quality and for testing the samples which are produced by the industry. Today, industry is not very much scared that some samples will be caught and the license would be cancelled, because we have a very poor infrastructure so far as implementation part is concerned. That's the reason the faith of the people is getting shaken, that the drug they are getting, whether that is of genuine quality or not. Problems arise right from the raw material. We have the quality parameters in place, and still we are not very confident whether those are really executed at the industrial level. We have more than 9,000 industrial houses in the country. There are very small scale industrial houses, large scale manufacturers, very few. They are not very much interested to make major investments because they can put the drug in the market just making certain proprietary products. They are not very keen to go into the classical products, which are the real strength of these traditional systems of medicines. Because it is very easy to produce a proprietary drug without undergoing all those biological evaluations or clinical trials or chemical evaluation. And industry is also shying away because there is no protection for the data they generate if they invest on R&D. Data generation protection is very essential, whereas IPR, intellectual property right, is protected in case of the drugs and pharmaceuticals. Our drugs and pharmaceutical industry has gone far off, whereas we are at a very lower level. 1/30th of the drugs and pharmaceuticals we are producing in the Indian systems of medicine, and the classical medicines are 1/10th of those. This situation has to change. And there is a dire need of strengthening the infrastructure for these things, which is very necessary. Now, coming to the research methodology, I think we should make the best use of the scientific approaches which are available to us. It's not that the credibility of the Indian systems of medicine is questionable. When there is a lot of scientific parameters are available, Dr. Sharma talked about holistic approach. 5 holistic approaches are necessary when we consider the traditional systems of medicine, and it is fully in consonance with the systems biology that we talk today. Systems biology has a holistic view, and Ayurvedic Unani concepts exactly fit into this. There is a lot of molecular biological advances which have taken place, and Dr. Rama has indicated the use of molecular targets, cell-based assays, enzyme-based assays for producing the biologically active activity data generation. Of course, that is not necessary as per Drugs Act, because we are convinced that the drugs which have been used since ages do not need any validation. But still, it is advisable because these drugs, whether they are single or compound drugs, they are multi-targeted drugs. We have multifactorial diseases, the metabolic disorder, the degenerative disorders, and the traditional systems of medicines are the right answer. It's not the modern medicine, where single drug and single target paradigm is in place. That is single bullet theory, which started at the time of antibiotics discovery, and that is continued with the discovery of modern drugs, single target based, but the diseases mostly are multi-target diseases, which are the problem diseases, and the traditional systems of medicines has the answer because they use multi-component ingredients. Therefore, there needs to be an interface between the modern medicine and the traditional medicine. Modern medicine is 200 years old, and our traditional medicines are thousands of years old. It's a question of adopting certain good things from the approaches which are being made in the modern system of medicine for the drug discovery and drug development. So far as formulations are concerned, I do understand that the clinical trials need to have a altogether different paradigm to choose for the traditional systems of medicine. There is a need to include into the protocols the prakriti of the individual, the drug profile, the dose regimen, the intervention, in between intervention, and so many other factors which are necessary from a holistic point of view. And those parameters are not considered necessary so far as the modern drug is concerned. Therefore, doing work in the traditional system of medicine is more complex, more sophisticated as compared to the single drug molecule discovery. Here we are dealing with, if we are dealing with a plant, it itself is a complex laboratory of innumerable substances, both the primary metabolites, secondary metabolites, and cellular material. And the traditional systems of medicine very much accept the holistic approach along with the cellular material like your churna. They contain the cellular material which is necessary along with the primary metabolites and secondary metabolites. We use decoctions, infusions, where we do not need the cellular material. And I think if we work on the single drugs, single plant drugs, we can really discover single drug is also a multi-component ingredient. Therefore, multi-targeted approaches in Vithania had been seen, Vithania ashwagandha, which is a Indian ginseng treatment for so many diseases, and it's a sort of panacea. This has been proven with science that it acts on multi-target in the human system. And a lot of chemistry has been done. RRL Jammu has done a lot of work. Gugulip is a simple example where a lot of scientific inputs have been made, and Gugulip was accepted at international level as a drug from Google. We have a whole category of Googles in the traditional systems of medicine, and only one segment has been investigated, that is anti-hyperlipidemic activity with Gugulip. And that's well standardized, well accepted, even in the modern system of medicine. Same is true with Salaki. For arthritis, Boswellia serrata, again work from RRL Jammu, where a lot of work was done in collaboration with the German scientists, and the mechanism of action has also been established. These are the kinds of plants which one can pick up. I can cite a number of examples of the single plant drugs where they have made a name from Ayurvedic origin. Picorrhiza kurua, kutki. Liver disorders, complex liver disorders. Now the fibrotic liver is a serious problem in the modern system of medicine. Surgical removal or replacement of the liver is the only answer. But there are innumerable plants which are present in our repository which can be investigated for hepatic disorders. I think there is a lot of differences so far as approach to the modern drug is concerned and the approach to the traditional systems of medicine is concerned. We should continue to do our traditional approach with the scientific bent of mind so that we make the best use of the science available. Now, the scientists from the other areas are very much keen to work with you so that you can develop some scientifically based protocols, both for quality, both for the procedures. You see, when we talk of the procedures of producing a drug, ras aushadhi, which are very potential drugs in Ayurveda. Balindu is here, who is a well-known clinician in Ayurveda, and he uses only ras aushadhis. But the process is so complex, and slight negligence in the procedure can create a lot of toxicity. The majority of the questions which are raised are with respect to such drugs, which are very potent, very good drugs, but because of the procedural non-uniformity, there is a toxicity. Because any metal which is consumed by human body has to have a toxicity if it is not properly treated. That is the Shodhana process or different processes which are known in Ayurveda. And that's where the science is necessary to be put into, and the standardization of all methodologies and trying to adopt certain modern engineering techniques, instrumental techniques, which can replace some of the classical methodologies. We are talking about the fire, which they are using, different kinds of woods are used. They give different temperatures. Why not to use an oven? Try to standardize at different levels. Now we have involved industry, and the Ayush Department of Ayush is funding even to the industry for creating the SOPs for such kind of drugs and also the quality parameters for bhasmas, for ras aushadhis. These are very crucial areas, very important areas. We are making beginning, but I think we should continue our endeavors. The scientific community is fully at the back of you, and we should work together in order to produce those drugs where there are gap areas and where there is no treatment available in the modern system of medicine. Those are the areas where we should work, both for the compound formulation as well as with the single drug. And I think I should not expand too much. And this is where I feel that as a person not knowing Ayurveda or Siddha or Unani at all, purely a pharmaceutical scientist having affiliation with these traditional medicines, would like to contribute lifetime contributions if we can lay down certain guidelines for evaluating these drugs. Those guidelines within the systems concept. If the clinical trials are to be designed, keeping in view the systems concept. If the procedures are to be laid down, keeping the concepts in mind. Having worked on Kashar Sutra, I know how the science is already built in the mechanism which is subscribed in the Shastras. So that is not a science. That is a science. It is the scientists who have to understand that science. We are not in a position to understand the scriptures, real in-depth knowledge which is imbibed in our scriptures. And with this approach, if we endeavor to work on our traditional systems of medicine and try to adopt whatever is good in the science, and I would say if we have to generate the biological data for efficacy, for validating efficacy, I would say let us depend upon the in vitro test systems rather than going the animal testing. Even for toxicity testing, I would not hesitate using the in vitro test systems, because now for seeing the safety of certain drugs, that they are not toxic, we can use human cell lines from liver, that is HepG2. Those are the cell lines which can be generated, and mass samples can be analyzed for safety. Same way for mutagenicity. The human-derived kidney cell lines, HK2, those are the cell lines which are important to see the mutagenicity if there is any of these drugs. Such kind of in vitro systems would be fast, give you the answer, very quick answers. Of course, subsequently from regulatory point of view, one can take the in vivo systems, but not from the testing point of view, because our drugs do not need too much of testing. Those are well tested on human beings. It's only to be doubly sure with the modern approaches whether we detect something which can otherwise damage the human system. With these words, I'm very thankful to the Department of Ayush, to CCRUM, and to CCRAS, who recognized my humble contributions and gave me this opportunity to give this brief